Okay. So I think I got all my wires hooked up. I don't know.
Don't want to? Not comfortable?
I don't like it. It's restricted. I don't feel like I can see as well. Yeah. I guess.
Given last week's environment.
I could just as easily die from cancer. That's right. Okay. Alright. Okay, so back on track. We left off, I believe we talked about the path of the electrons, right? And we talked about how the electrons get pumped across the membrane, and that Peter Mitchell got the Nobel Prize for this in 1978.
And gave no credit to
Yeah, don't give no credit to the students. So, essentially, let me just zoom in the camera. No, not out. All right. So, we have complex 1, and then complex 2, and coenzyme Q, 3, thank you, and cytochrome C, and then 4, and this is the electron transport chain. So what happens is NADH goes up to complex 1. Complex 1 pulls 2 electrons off of it. The electrons go through 1. Let me use a different color. So the electrons go through 1, they skip 2, through CoQ, through 3, through site C, through 4, and then they, what's the ultimate electron acceptor?
Oxygen.
Yeah. And that combines with hydrogen, and that forms water. All right, so 2 electrons go through here. How many protons do you think would be attracted to 2 negatively charged electrons?
2.
And so that would be right. So 2 protons get pumped out through complex 1. 2 protons get pumped out through complex 2. 2 protons get pumped out through complex 4. So for every NADH, we pump 6 protons. Is that the same for FADH2? What's the path that FADH2 takes? Yeah. So,
Gives 1.
right, so FADH2, let me give it a So FADH2 holds its electrons too well. Complex 1 can't pull those off, but complex 2 can, and it does, and then the electrons follow the same path. So, how many protons get pumped out by FADH2?
4.
Good. All right. Any questions about that? So this is a one-way gated transporter. So once the protons go out, they can't go back through that same hole. There we got it? All right. So that brings us to how is ATP made? Because none of this is made ATP, right? All it's done is pump protons. That's the whole purpose of the electron transport chain. Well, it turns out that there's another separate part of this. These have to be in this order, right? But there's another thing called complex 5, and it can be anywhere. So So complex 5 is also known as ATP synthase, and before they knew what it was, what it did, they called it an EP, an elementary particle, because they didn't really know what it was. So I'll just show you. What? Well, I don't know. We'll just ignore it and see what happens. Okay, so this is an electron micrograph of the inside of a mitochondria. So what kind of electron microscope took this picture? Scanning or transmitting? Very good. And so you can see these little dots here. Those aren't ribosomes because they're in the inside part, right? The ribosomes are on the outside, not of the mitochondria, but of the, what? Endoplasmic reticulum, good. So this is on the inside of the mitochondria. What do we call the area inside the mitochondria? The matrix. Excellent. Here's another electron micrograph of complex 5. And it's labeled EP because they didn't know what it did. And this is the late 80s, just to give you an idea. So what happens is that we have all these hydrogens that are pumped across this membrane. This is the intermembrane space. And so here's our membrane. And this is the matrix. And what happens is the protons go through here and they cause this to rotate. And when it does, there's a drive shaft in it, just like in a car, a rear-wheel drive car, and that causes, this is called F0. This part right here is called F0, and this part right here is called F1. So F0 turns and causes F1 to be affected by this rotation here. So I'll show you guys how that works. This is the setup that I just drew. So this is F0, lies in the membrane, and then this is the gamma subunit. You don't have to know this, but this is the drive shaft, and this is made up of two different subunits, an alpha and a beta, and they rotate. So there's actually 3 core enzymes here that are making ATP on each side of it. Let's see if this works. Yeah, that's what I thought. So I'm trying to load up these videos individually. Okay. So this is a side view. You guys are looking on F0 like this. And so there's the channel that it makes. Kind of looks like that. And there's two parts to it. There's an outside and an inside. So there's two wheels. One's on the outside, one's on the inside. These are made out of amino acids and they can rotate. So we have the blue dots here are the hydrogens that are located out here, right? In the center is negatively charged R group. And so what does negatives attract? So the hydrogens are attracted to this and they'll come down and they'll bond. You guys see that?
Well, how come like in the beginning they're coming in with hydrogen now?
These are hydrogens. So there's in this wheel, there are negative charges. And this spins. And then on this protein, on the outside part, there's a positively charged molecule. All right, so here's the hydrogens that are positively charged. They're going to be bound to the negative, right? Do we see that? And then, so what happens when you have two positives? So here's this fixed positive, and now there's a positive attached to it. What does it do? It repels, so it starts pushing it. And as it comes around, it's still bound to the hydrogen. So the rotational force of this forces this to get pushed away because the two positives are going to repel and it pushes it down inside. Now it's a negative, so what's the negative and the positive do? They're attracted. So that causes this to spin around. So your electricity is alternating current. And it works almost the same way. So this is identical to how your, say, vacuum cleaner motor works. Except living things have been doing this for, however long you believe living things have been around. But I believe for millions and millions of years. So that's pretty amazing. It's just a tiny motor. And so that motor drives this. This is the F1. We can get it to open. Not you. Okay, so this is, if you're standing underneath this thing and you're looking up, right? So remember there's subunits in this. So we have the alpha subunit and the beta subunit, and then this is the gamma drive shaft. So you can see that here, except, one of these is yellow, and one of these is red, and then this is gray. In that image. So the drive shaft, this gray and blue part right here is turning. And it's turning because F0 is turning. And it's attached to it. And then how does that affect the A and the B subunit, the alpha and the beta subunits? What are they doing? They're changing shape, right? In fact, what they're doing is they're opening, and then they're putting ADP and a phosphate in there, and then what are they doing? Closing. So it's literally just like if I gave you the ADP molecule and a phosphate and said, put those together. What would you do? You push them together. That's what it's doing. It's physically pushing the molecules together to cause them to form the bonds. And so that's how ATP is created. And it's doing it simultaneously on each side of this enzyme, ATP synthase, which is where it got its name. What do you think it does? It synthesizes ATP. Do you guys get it?
That's all in complex.
5.
Yeah. So F0 is rotating from the hydrogens coming through there. And this is physically changing shape to make ADP and phosphates into ATP. So these are some other videos which I'm sure will not work. This is just another model that shows you the rotation. You guys can maybe be able to get it to work if you have like a word viewer that's earlier than 2010, which is what this one is. Oh, it's going to work for a second. This is just showing that what goes in is ADP and phosphates, and then what comes out is, what do you think? ATP. So here's another sort of a drawing. I mean, it's much easier to visualize if you can see it in action, but we can go over it in one of these artist renditions or renderings of it. So here's the gamma subunit, the one that spins around, right, the drive shaft, and that's caused by F0 spinning. And so let's just start here. So if we look at this first subunit, so this is the alpha and the beta put together, so it's called the beta one. So it's open, right? And in that open position, it can bind ADP. So this thing rotates, it changes its shape, and it binds a phosphate in there, and now it's closed, the reaction occurs, it makes ATP, and then that turns again, and then it opens, and then what does it release? ATP, and now it's reset to take in another ADP and a phosphate. So it's doing this. And each side is at a different point. So when this one's binding it, this one's causing the reaction to occur. And this one is starting to push them together. Yeah. So all three sides are reacting. So one spin can create 3 ATP. That make sense? So these dudes, Walker and Boyer, we'll give those guys a couple of Nobel Prizes for chemistry that they got in 1997. So $1 million, actually they got 10 million. And 2 chunks of gold.
How long did it take them to figure that out?
About 7 or 8 years.
It's worth $10 million.
Yeah, I'd take it. Okay, so I'm going to show you guys another video. So there was a Nobel Prize awarded yesterday. You guys know what it was for? Nobody pays attention to that but me. I have it on my Twitter feed, Nobel Prize. Yeah. Just kidding. So the Nobel Prize was awarded for a special microscope called a laser confocal microscope that was developed so that you can visualize things at a higher magnification than 1000x, because the wavelengths of lasers, you can focus those rather than the scattering of light. And so they won the Nobel Prize for chemistry yesterday for that invention. So this, what I'm showing you, the reason I'm telling you this, isn't to kind of update you on science, although you now have to, but it's to let you know that this is the same technology that's being used on this video. So what they've done is they've taken a fluorescent tag, and it's sort of like a long flag, right? Like maybe like something you'd see at a football game at halftime for the band or something. And so you can see this because it's a fluorescent tag, and it's gonna spin, and it's gonna be in real time, so that you can actually see this ATP synthase in action. So the first clip, the membrane has been punctured. So what would happen then if we punctured this membrane? Would you expect this to rotate?
No.
It shouldn't, right? Because the hydrogens aren't forced to go in here. Now they can leak back down. So the first part of the video, it won't spin very much. There's not very many hydrogens going. The second part of the video is this has been restored, and so you should see this thing spinning like crazy. And every time it spins around, it's making 3 ATP. And remember, you have, hundreds to thousands of mitochondria per cell. You have millions of ATP synthase per mitochondria. So you literally have trillions of these things all spinning around right now inside your body in every single cell. This So you can see that this little flag, it's not really moving very much at all. It doesn't have any rotation because there's no hydrogens that are available to go through it. The screen will black out, and then you'll see it closed off. And one of my students in the other class were like, "That's the best resolution you can get." I mean, this is literally just a few molecules in size, so that's pretty good. And so you can see that it's rotating, right? And while it's doing that, it's producing ATP. And
That's real time.
That's in real time, yep.
Okay, cool.
Pretty cool, right? Or no?
That's cool.
Okay, so here's some of the math that you guys are going to need to know for the test, because the extra credit, remember the extra credit is that I wanted you guys to look up human diseases that are caused by deficiency in ATP, or you could look up other organisms, but I prefer humans, so I ask you to do that instead. Because there are lots of diseases that scientists think are caused by deficiencies in ATP. One of the biggest ones is what happens when you can't make enough ATP, or you don't make as much ATP? What happens to you?
Tired.
You're tired, what else?
Sleep.
Yeah, what else?
Weak.
What else needs ATP?
Brain.
Yeah, so you forget stuff?
Ah.
So what am I describing? Getting tired and forgetting and it's not well, that's one disease, but what else? It happens to everyone. It happens to everyone.
Old age.
Aging, right? So aging is caused by, I didn't hear you say that, but aging is caused by breaking down of the mitochondria as we get older. Do you guys know how much money was spent on anti-aging products last year?
1 trillion.
It's higher than that.
3 trillion?
It's higher than that.
20 trillion?
It's the entire United States debt. Like $16 trillion was spent on anti-aging products worldwide last year. So if you can figure out how to stop this, right? It'd be a lot of money. All right, so that's why I want you guys to be able to calculate this, because you need to know what the effects are based on different conditions that may occur. And it's not just natural things, but chemicals can affect the electron transport chain too. So let's go back over this so you guys know that we can produce 6 protons per NADH and 4 per FADH2. So how many protons can we pump total for NADH? In other words, how many NADHs have we made from one molecule of glucose so far?
How many what?
Molecules of NADH have we made so far
10.
from glucose? And the correct answer is 10. Now, why is it 10? Where do we make them? You're going to need to know this for the test. Remember, I told you guys need to know what goes in and what comes out. When do these come out? When does NADH come out? 2 glycolysis. Excellent. So how many in the Krebs cycle? 6. So we're missing 2. Where's the other 2? Pyruvate gets converted into acetyl-CoA. So that's 10. So how many protons do we have then? Total. 10 times 6 is good. And then, remember I told you that it takes 6 protons to spin that whole thing, so so we can make 3 per turn, so that means 2 protons make an ATP. So we just divide that by 2, and how many ATPs do we get? So 30 ATPs are produced from 10 NADHs. And then how many FADH2s do we make? 2 is correct. You got that off the screen, didn't you? Yeah. And so we got 4 protons, so 2 times 4 is 8 total. We divide that by 2, and how many ATPs? So that gives a total of 34 ATPs from electron transport. But I told you guys we made 36, so did I lie to you? Where's the 2 ones that were short?
They have given back from Krebs cycle.
Yeah. No, we still have 2 that we got from the Krebs cycle, so we get 36 ATP. I told you bacteria make 38. And why is the difference? Yeah, because we have to pay 2. This is where we lost the 2. It cost us to put the pyruvate into the mitochondria. All right, do we follow that? Okay, so now we're going to play a little game. So, question 1. How efficient is our organisms in converting the energy from glucose into the energy of ATP? Remember, what's the second law of thermodynamics? Every reaction causes disorder, right? And in our world, that disorder is energy lost as heat, because heat is a random disorder of energy. So you're always going to lose some energy to heat, whether it's driving a car or burning glucose in your body. So the question is, how much energy have we lost? So what did we start with? How much energy is in glucose that I told you the delta G of glucose is? No.
Right there on the screen.
Yeah, shut it.
686 kilocalories per mole, right? And so remember that in the lab, the ATP's value is 7.3 kilocalories per mole, right? So how many ATP did we make? $36. And so what's the total kilocalories that we get out of this? hope you guys either bring your calculator to the test or you like to do a lot of long,
262.8.
thank you. 260,
2.8.
so what percent efficient is it? We divide that by,
38.3 or approximately 40%.
okay, well, you read that off the board, but let's just check my math, because I could be wrong, right? So that should be 0.38, roughly.
30903.8.
Yeah, good enough. So we're about, if we take the data from the lab, we're about 38% efficient in converting the energy from glucose to the energy of ATP. That means that we lose about 60% of it to that second law of thermodynamics. But in reality, and remember I told you guys you want a Nobel Prize, figure this one out because no one has. Inside the cell, it really can make 13 kilocalories per ATP is the value. So what's 13 times 36? $468,
Yep.
and then we divide that by 686, and we should get somewhere around
70%.
70%. So that's massively efficient. I mean, if you look at like the most efficient gasoline engine, that means that gasoline's ability to convert the energy in gasoline to the energy and kinetic energy of moving forward and miles per gallon, you're looking at maybe 18% efficient. So you are, 4 times more efficient essentially than the most efficient gasoline engine that there is. That's really good.
So the Nissan Leaf gets about 55 miles per gallon.
That's electric equivalent. It depends on how much you pay for electricity. I have a Volt, right? So I calculate that all the time, which is what you should have bought.
I get 36 miles per gallon highway. I'm good.
You know what I get? 182.
Whatever. I gotta drive to California.
And I'm gonna put some solar panels on my roof and get it for free.
You're not gonna get it for free.
They got you,
You have
huh?
to pay for the solar panel.
Yeah, and then they want a lot more money to install it, right? But I think I've outsmarted them because I'm gonna buy inverter panels so I don't electrocute myself.
But you know that you can also sell your
We'll see.
electricity to the electric
If I don't show up next, and over like after Thanksgiving, it's probably because I died from electrocuting myself putting on solar panels.
You know that if you generate more electricity than you use, you can sell it to the electric company.
Right, it'll make your meter run backwards.
Yeah.
Yeah,
Wow.
that would cover my whole roof though, because I need about 15 kilowatt hours.
I'm sorry, what is it? Where's that 13 come from?
So that's the value that was calculated from inside the cell. So for some reason, inside the cell, you can get 13 kilocalories per mole from ATP, but in the lab, what we can measure is 7.3. So there's something
Okay.
weird going on that causes it to be more efficient in the cell than outside of it. And nobody knows why. But it happens, and I promise you, whoever figures that out will win a Nobel Prize. Guaranteed. All right. And all this stuff is important because there's a whole sector of biology called bioengineering, which is basically looking at biological systems and creating mechanical processes. That's where solar panels came from. All right, so let's continue our game. What would you expect to happen if complex 1 was faulty? In other words, this is broken. How many ATPs could you make? I got 2. I got 8, 4. Anyone else? Yeah. Want to throw a 5 or 6 in there? 6? You got a 6 in there?
I'll throw a 6 in
So the correct answer is
there.
6.
Six.
So how many ATPs? So Remember, FADH2 doesn't need complex 1, right? So we can still pump protons from FADH2. How many ATPs can we make from the FADH2s? 4. And then how many did we get from the Krebs cycle? So 4 plus 2 is 6. So the correct answer is 6. Excellent. So, okay, here's another one. Complex 2 is involved in the Krebs cycle electron transport. Well, what would happen if succinate was not available to the cell or it couldn't produce succinate? Just a reminder, succinate is part of the Krebs cycle, right?
It doesn't work.
So if it can't go around the circle, what happens to the Krebs cycle?
Stops.
It stops. And so what happens if the Krebs cycle stops? How many ATP can you make? Where do you get those two? Glycolysis, right? Glycolysis. That almost sounds like a disease. Yeah, so that's right. 2 from glycolysis because the Krebs cycle is completely shut down. However, you could also make more because what else do you make in glycolysis? If you're breathing still. Remember you make 2 NADHs?
Higher.
We can use those. And then where else do you make NADHs? You could still convert pyruvate into acetyl-CoA. So now you have 4 NADHs that you can put in this, right? 2 from glycolysis, 2 from the production of acetyl-CoA. So if we have 4 NADHs, how many ATPs will we get from that?
4 NADH. 18. Oh no, 18 divided by just 9 ATP. Hold on.
Okay, so we have 4 NADHs. How many protons do they pump? No. 2, 4, 6. So that's 24 protons. And how many do we need?
12.
That makes 12 ATP. And then
Whoever said 18 threw me off, that's why.
we lose the 2 ATP because we still have to push the pyruvate into the mitochondria. So the correct answer is 12. What if complex 2 could work to make succinate, but it couldn't oxidize? That's a big word. What does that mean? Lose electrons, right? So it couldn't take the electrons off of FADH2. How many ATP would you get? This is actually common. This is a common occurrence in individuals with Alzheimer's.
Like what happens to what?
If complex 2, remember complex 2 is the only one that's involved in the Krebs cycle and electron transport. So the Krebs cycle's fine, but the electron transport part doesn't work. This one works now. So how many ATP can we make? So everything's the same except we don't get those. So how many can we make? 32. That's correct. So this is common in Alzheimer's and Parkinson's individuals. What, cyanide and carbon monoxide. So we talked a little bit about that. Cyanide binds right here. It would be the same as me cutting the electrical cord on your vacuum cleaner. What's the effect of that?
Pure power.
Electrons can't flow. This whole thing shuts down, right? Everything behind it won't work. So what's the end result? How many ATP? It's not 0.
2?
Yeah, it's 2. Where do you get those 2? So
Glycolysis.
glycolysis, because
Glycosis.
now glycosis. So cyanide binds covalently, so this isn't reversible. So if someone came in the hospital and had cyanide poisoning, what would you tell them? Sorry,
Give them oxygen.
it wouldn't work. They're done. So, yeah, so like in California, they use cyanide in the gas chamber to execute people. Once that starts, you can't reverse it, right? Once you're down that path, you're done. You can't pull them out and go, oh, I'm sorry, I made a mistake. We found out from DNA evidence that you didn't do it. It's too late.
Kill them in gas chamber. Kill them in gas chamber.
Yeah, in California, that's what they do.
How fast do you go?
10 minutes. How long can you hold your breath? Because it's the same amount of ATPs, right? So being poisoned with cyanide is the same effect as holding your breath.
Painful?
I don't know, hold your breath. Is it painful?
Yeah.
It's a terrible way to execute someone.
Well, when I lived in North Carolina, Jesse Helms wanted to take a flatbed truck around with an electric chair on it and have public executions. Wasn't kidding. Okay, so carbon monoxide binds to the same place, except it binds ionically. So is that reversible? Yeah. So the two molecules, exact same pathway, except one is reversible and one isn't. What would happen if the intermembrane leaked? How many ATPs could you make?
If it leaked,
Yeah,
0.
two.
Only in the beginning.
From where?
Glycolysis.
Yes. Because that would be the same effect as shutting this down, right? You wouldn't be able to get the protons to go through there. Make sure you guys know the parts of the electron transport chain. I promise you I'm going to ask you, what path does this take? So what path does NADH take?
Complex 1, 2,
1,
three.
go Q, 3, site C, 4, and on to oxygen. What path does FADH2 take?
Complex 2, CQ, 3.
Oxygen, good. So what would happen, so all the scenarios we've been going through, I expect you guys that you should be able to tell me what would be the effect. So let's play another one that's not on there. What would happen if there was another chemical that bound here and shut down the electron transport chain? How many ATPs could you make?
6.
6, because you would get 4 from FAD and 2 from glycolysis. Excellent. What if there was a compound that bound here? How many would you get?
It stops. Complex 2.
It's at CoQ, so it starts at CoQ.
So 2 right here.
Only 2.
Right. You guys get it? Good. All right. So make sure you know the parts of the mitochondria. I'm going to ask you questions like, where is F1 located? It's in the matrix. Where's F0 located? In the inner membrane. Where's the electron transport chain located? It's in the inner membrane. Where is the Krebs cycle located?
Matrix.
It's in the matrix. Where does glycolysis occur? The cytoplasm. Good. Yeah, I can't think of anyone off the top of my head right now. Oh, where are the protons built up at? In the intermembrane space, right? Or inner membrane space. What part of the electron transport is also part of the Krebs? Complex 2. I don't care that you know Peter Mitchell is the one that did that. But you should know how it works, right? So are electrons driving this? If I ask you on the test, are electrons driving this, true or false?
Yes. Because positive,
No
negative.
electrons are even touching this. What's driving this? Protons. The electrons are only pumping protons. That's all they do.
I thought there was like a positive and negative effect.
That's due to protons, not electrons. It's only due to protons. This thing would not spin. It wouldn't matter how many electrons you threw in this thing. It's not going to spin unless it's given protons. So that's how it works. No protons, no ATP. Well, some, but not much. All right, make sure you know the parts of, you don't have to know all the parts, just where they're located. So make sure that you know that there's two parts to ATP synthase. F0, what is F0's function?
Spins.
It spins. That's all you really need to know. And then F1, what's its function?
Bind ATP.
It smashes the ADP and the phosphates together to make ATP. What drives this? Yeah, protons, that's it. It's not anything else but protons. I already said that. Why is it better to golf with like golf clubs with graphite shafts than steel shafts in an electrical storm?
Because plastic doesn't conduct electricity.
Right, so the point is different things have different affinity for electrons. So if this was less electronegative than that, would the electrons go this way? It would stop, right? So it's important that these are in this specific order in the electron transport chain, or they wouldn't be able to pass on the electrons because they each have a higher electronegativity. What's the ultimate electron acceptor? Good. Any questions about that? Right. So you're going to see this again in chapter 10. So if you don't learn it now, you're going to have to learn it again when we do photosynthesis, because photosynthesis works almost the exact same way. In fact, in the chloroplast, there are ATP synthases too. What do you think they do? Yeah, in the chloroplast, there's an ATP synthase. What do you think it does? It makes ATP. It's exactly right. How do you think it does it?
Protons.
It uses protons. How do the protons get across the membrane? Electrons. Same thing, right? So plants use this for photosynthesis. You're going to see it again. Okay, so what if you're not breathing? All this is if you're breathing, but there are instances where, let's say you're running or you're being chased by a bear, you can only breathe in so much air, right? The first thing you do when you start running is what? You breathe faster, right? And then your body, you're burning more energy, and so your core temperature goes up. So you start sweating to cool yourself down. All this is byproducts of the second law of thermodynamics. So, all these chemical reactions are going on, but your lung, you can only breathe so fast, right? You can only take in so much oxygen. There's a limit. And what happens when you reach that limit? Do you stop making energy? You can do fermentation, right? Which makes only 2 ATP. And so, you guys know Lance Armstrong? You know who that is? Right? So they caught him cheating, but he wasn't using steroids or anything like that. Do you know what he was doing? He was blood doping. You know what that means?
Hypertuber.
No. So what he was doing is he would take his blood out, right, before a race, a few weeks before a race. So his body's regenerating, replacing that blood, just like if you guys went and donated blood, right? After a few days that you would be fine. Your blood volume would go back. So before a race, what he would do is reintroduce another pint or so or 2 of blood into his body. What does that do for him? That gives him the ability to load more oxygen so that he doesn't get into fermentation, because once you start fermenting, your muscles start cramping. So people are riding up the hills, right, that weren't doping, And they're like, oh, I'm going into lactic acid fermentation because I can't breathe anymore, and my muscles are cramping. And there goes Lance Armstrong. Woo!
You ever seen that show on Animal Planet, or was it, I don't know which like scientific channel it has? It's called Superhumans, where they had this guy that can run until he falls asleep, pretty much, and doesn't fatigue, doesn't tire, just keeps running.
As
running,
long as
running.
you run at a pace that you can breathe in enough oxygen, you can burn it until you run out of fuel. The problem is if you're exceeding that capacity.
Yeah, but he doesn't get tired. Like it's
Well,
off.
do you know what organism can run the longest? Humans. Yeah. So that doesn't surprise me at all. But once he started going into fermentation, he would have to stop running, or he
Why
would die.
doesn't stop running?
Well, he's not going into fermentation because he's running too slow. You got to tell him to pick it up, right? well, let's release some tigers behind him, and then let's see
How long?
if he goes into fermentation or not, because he will. I
Cheetahs
promise
can only
you.
sprint for a minute, or not even 35 seconds.
Yeah, so we need another organism that can chase him for a longer time period. Anyway, back to fermentation. So there's two kinds of fermentation. Remember I told you guys glycolysis happens, you might as well get the bumper sticker. So here's glycolysis. We're not breathing oxygen. It produces pyruvate just like before, if you're breathing. Except pyruvate doesn't get put into the mitochondria. It goes and gets converted into acetaldehyde. This is alcohol fermentation. And then the NADHs that we make in glycolysis, right, same thing, are used to convert this, which is a sort of a toxic product, into ethanol, which is not quite as toxic. So we don't,
It's 9:30.
yeah, so it's about an hour. I can't believe that nobody else has this issue. And it's blinking, so I can't do anything about it. But, so I turned your attention to this one, which I'm sure will go out in a minute or two.
Goes down 5 minutes.
Whatever. So humans can't do this, right? Because if we're running away, if we're getting chased by a bear or whatever, and you started doing alcohol fermentation, what would happen? Yeah, and then you'd be like, oh, hey, Mr. Bear, can I hug you? You want to eat my face off? I don't care, So humans and animals don't do this, but yeast do. That's how beer and wine's made, right? So you put sugars in, and you put the yeast in, and guess what they make? They make alcohol and what else? Carbon dioxide. It's easy. And then we do lactic acid fermentation, which is very similar. So we produce pyruvate, but that pyruvate is converted into lactate, which is lactic acid, which is sort of toxic to your muscles, so it causes them to cramp up. And there's another system, which you guys will learn in anatomy and physiology, which diffuses that, converts it into another product, and so on and so forth. Right. So, but I don't want to, we have other junk that we need to talk about, so we're not going to go down that path. Everybody understand the difference between fermentation and respiration? And out of these we get 2 ATP. Alright, so this, I showed you guys the metabolism diagram with all those dots and all the different enzymes. So you know that all we've talked about is glucose, but you know that you can use other sugars, right? You can use fructose, you can use lactose, you don't even have to use sugars, right? You could use other things, like what else on your nutritional label can you use for energy? Fats, proteins. Alcohol is another one, right? You can actually use that as an energy source too. So all of these things all paths lead to electron transport. No matter what you're eating, all of that gets broken down and put into this glycolysis, Krebs, and electron transport pathway at different places. So depending on what kind of amino acid it is, it'll get shunted into here or at acetyl-CoA or directly into the Krebs cycle. Some sugars bypass the first few steps of glycolysis and enter in the glyceraldehyde 3-phosphate. The fats, remember, they're made out of glycerol and fatty acids, so the glycerol can go in here and be converted to glyceraldehyde 3-phosphate, or the fatty acid tails can go and get put into the reaction pathway, the metabolic pathway at acetyl-CoA. So the point is, I don't want you to memorize all this, but the point is that no matter what you're eating, right, it's going through this pathway in some way, shape, or form. All I want you guys to know is the glucose one.
So the bypasses glycolysis.
Some things do, some things enter in the middle of it, some things enter early in it. It really depends. I mean, there's sugars that we've never even talked about that you probably don't even know that you can use, like arabinose. You can use as an energy source. Did you know that? You ever heard of it? Yeah. So there's lots of sugars that we haven't talked about that you can use as an energy source. This is just a small part of the bigger picture. You know, you could have a whole class on metabolism, and there is one. It's called biochemistry. And remember that we can synthesize other things, right? So if you eat a lot of carbs, you store that energy as fat, right? If you don't run out of amino acids, you can produce them. And all of these parts are run in some way, shape, or form through that metabolic pathway that I showed you. So that metabolic pathway is really important, not only for breaking down stuff, but what else? Building it up. What do we call it if we're breaking it back down? Catabolic. And if we're building it up, it's anabolic. All right, that's it.
Are we doing a review on Tuesday?
Yes. So that covers all the chapters, but I'm going to need to spend the next 10 minutes or whatever going over photosynthesis because your third test is going to be, we're only going to have 4 lectures before that test since I pushed the other test back. And I can't push the other test back because then you'd have to come in on Thanksgiving and that would piss people off.
I can come in. I don't have anything before.
Are the
What?
online episodes due the next day?
Okay, you can come over.
The playlist is gonna be the 6th.
Yeah, I moved all the dates to the 6th. I did that while you guys were out to Panda Express between the wherever you guys did between the lab and lecture.
How do you know?
Oh, that's right.
Dr. Lewis, how do you know we go Panda Express?
What?
How do you know?
How do I know? They told me.
Oh.
I spied on you. I followed you around. No.
I thought we should start out at
It's scary.
We're going to be talking.
Okay. So this chapter, and we got both screens up. So this chapter is on photosynthesis. This won't be on this test, but it will be on your third test. Chapter 10. So chapter 10, 12, and 13 will be on the test that you're going to have right before Thanksgiving. All right, so you guys know the reaction for photosynthesis. It's the reverse of respiration. So plants take in carbon dioxide and water, and then they produce oxygen and glucose. And then they store their glucose in amyloplast, right, as carbs. And then they use that to make other things, like if they need to make DNA nucleotides or RNA nucleotides or amino acids or anything else. They use those sugars as a basic building block source to do that. It's a little harder for plants because they don't just go around eating amino acids, right? You don't see a plant running around eating people or anything like that.
Told you 5 minutes.
So anyway, the slide, it's just showing the formula for photosynthesis. All right, so when we talk about plants, we're talking about, and you guys already learned this word, right? So autotrophs versus heterotrophs, and we talked about this in chapter 1. So if you're autotrophic, what do? Make your own food, right? And if you're heterotrophic,
Or sunlight.
then you rely on other organisms to make your own, for your energy source, right? So what are we? Can you guys go outside and photosynthesize and just be like, I'm not hungry, I'm just gonna go lay in the sun and, no? So we're heterotrophs, plants are autotrophs. There's another class of organisms, well, the autotrophs can get split into what we call photoautotrophs. And what's photo? Light. So they use light. And then there's chemiautotrophs, and they use chemicals. So scientists didn't know this. Like, we had already put a man on the moon before we knew what was at the bottom of our own ocean. And so, you know, in the 70s, scientists developed a method. I can turn it back on. Developed a method to go and explore these deep ocean areas, and what they found down there was pretty surprising. So they found these what we call hydrothermal vents. So, or they're also known as black smokers, and they had this whole ecosystem that was living down there. And the source of that ecosystem were these bacteria that could take hydrogen sulfide and convert that through the same sort of mechanism as the glycolysis into the energy of ATP. And so organisms were living off the bacteria, and you had this whole ecosystem. And it's not based on sunlight, right? It's based on chemicals. So this is pretty much the impetus of us going to Europa, because even though there's sheets of ice on top of there, the core is just like the Earth's core, right? So it's radioactive, and it produces heat. And so you may have these hydrothermal vents at the bottom of Europa, and that means that if life can live here on Earth there, then it could be on other planets as well. All right, so I just want you to know that not all things that make their own food have to use sunlight. Chloroplasts are the organelles that we already covered in chapter 6. Remember, and chloroplasts have how many membranes? 2, an inner and an outer, and then inside those membranes are, what do we call those things? No? Well, yeah, stacks of the things are called grana, but what are the individual things called? Thylakoids, and thylakoids are really the site of photosynthesis. So the chlorophyll is found in the membrane of the thylakoids, which again is from chapter 6. And plants have stomata, which is basically like you guys have like only like 3 ways to get air in your 2 nostrils and a mouth. But plants have literally thousands of nostril mouths, whatever you want to call them. So they can do gas exchange pretty easily and a lot better than you can, a lot more. They transport things through what we call veins. You don't really need to know that, I don't care. Let's just focus on the cells, right? So here's the inside of a cell. If you guys looked at the Elodia under the microscope, you would have seen this. Here's the thylakoids. This is the site of photosynthesis. This is a cross section of a thylakoid. What kind of electron microscope took this image? Scanning only looks at the outside. This is the inside of a thylakoid. So what is it? Transmitting. Excellent. I promise you I'm going to put a picture of an electron micrograph on the test. Well, I can't promise. I'm 95% sure that I will. I'm thinking about it.
Depending on the printer.
Yeah. So what's the inside part of the chloroplast called? No? Stroma. Where's the DNA in the chloroplast?
Stroma.
Good. The thylakoids are located in the stroma as well, right? So in photosynthesis, there's two reactions. There's a reaction that we call the light reaction, and then the other one that's called the dark reaction because it's evil. It's called the dark reaction because it doesn't require light. Right? The light reaction requires light. Dark reaction doesn't. It's also known as the Calvin cycle. Kind of like the Krebs cycle is also known as the citric acid cycle. Right? So it's named after Dr. Citric Acid. Fine. All right, so the light reaction takes light and it takes water. It splits water. And the whole point of this is to produce ATP and NADPH. What do you think that is? You seen a molecule like that before?
NADH with like phosphate.
Right. That's exactly what it is. NADH with a phosphate. What was the role of NADH?
Transporting.
Transport electrons, and guess what? Plants do that too. And then ATP is created, just like we showed, using ATP synthase. So these molecules feed the energy for the dark reaction or the Calvin cycle, which is also known as the carbon fixation cycle because this was where it takes carbon dioxide and produces sugars. The sugar that comes out of here is glyceraldehyde 3-phosphate. Remember that sugar in the middle that got split in half during glycolysis? And we had two of those halfway through glycolysis? Well, go back and look. That's what plants make. Do you have a question?
It's about the diagram, but I get
You don't like my diagram?
No, I like
This isn't mine, it's from your book.
I'm just saying that it's like, so I'm seeing the CO2 come in, and the CH2O comes out.
Alright, so we'll go over each one of the steps individually. Alright, so the light reaction occurs in the thylakoids, right? In the chlorophyll, the magnesium molecules actually react here. Reduces, what does that word mean? Right, so something that reduces is taking on the electron, right? So it reduces NAB plus to NADPH, which is just the same as NADH, except it has, what's the P stand for, you think? Has a phosphate on it, good. Produces oxygen as a byproduct of splitting water. That's the same thing that happened tonight when we did the gel, right? When we ran the electricity through the water, it produced hydrogen gas. You guys know the cars, the new hybrid cars, or not hybrid cars, but they're hydrogen powered, so they're fuel cell cars. They run on hydrogen. You can make your own hydrogen. All you have to do is run electricity through water. You could do it in your house. Yeah, it's explosive, so it's a little dangerous. Just as dangerous as putting on solar panels by yourself. 800 or 900 volts is enough to kill you in a second, but you can make your own just the way that we did in the lab. And then you wouldn't have to go and buy it at a filling station. But the idea of fuel cells came from cellular respiration, because what are you doing to make energy? Combining hydrogen and oxygen. And what's the byproduct? Water. So that's what those cars make. Anyway, it generates ATP by adding a phosphate to ADP, and it does it exactly the same way as we did in electron transport. And I'm out of time. So, have a happy and safe Halloween. Have a good weekend, and I'll see you guys on Tuesday, where we'll do a review. I might lecture for 15 minutes, and then we'll spend the last hour reviewing for the exam on Thursday. All right? Good? All right. I have a daughter and a son. She is going to be, I don't know, some form of princesses. And my son is gonna be, I think he's a Ninja Turtle, but don't ask me which one because I don't know them. I never watched that show, but my wife knows all of them.
How about you? What? What? Those suits, like with nothing on them, like